Hello students, today we are going to discuss detail on genome editing. Genome is the genetic material of an organism. It consisting of DNA, RNA. Normally RNA in RNA viruses. In prokaryotic system, with the exception of viruses as well as in eukaryotes, we contain genetic material DNA as DNA is present. The genome includes both the genes, that is coding and as well as non-coding region of the DNA as genetic material, and further the circular DNA is found in the case of mitochondria as well as chloroplast. The term genome was created in the year 1920 by Hans Winkler. He is a professor at the University of Hamburg, Germany. We should know what is genome editing. Before going to the detail, the concept of genome editing is not limited to genes. It also making alteration in the non-coding regions of the genome and epigenomes. What are the targeted interventions? We have to alter the structural or functional characteristics, just like color or number of blooming in flowers, some disease traits in animals and plants, all these things. Already we discussed how to edit, how to treat our genetic disorders. The approaches of genome editing is called as reverse genetics. That is, the mistake which is found in the case of the gene is reversed to normal, so it is referred as a reverse genetic. Among the key requirements of reverse genetic analysis is the ability to modify the DNA sequence of the target organism. The genome editing was selected by natural methods as the 2011 year. That is, the CRISPR cas system was selected by 2015. It's a breakthrough year. Way to produce a disease-resistant plants. The pathogen produces virulence factor. How can we neutralize means by suppressing the virulence function. And plants have susceptibility genes where we can go for genome editing. Many pathogen attack trap. Introduction of the novel or genes and stacking multiple genes at the preferred site can prevent the infection from the pathogen. The many pathogens are defeated by many R genes. So by restoring the defeated R genes, where we can produce disease-resistant plants. So methods for the various genome manipulation: classical breeding, targeted genome editing, and transgenic approach. An alternative to both classical plant breeding and transgenic approach, this is genome editing is utilized. Here we can see some variation in the case of our various plant products. That is cross breeding or conventional breeding method, and genetic modification methods, and last one is gene editing method. Here is a list of comparison of modern gene editing technology which is available in the case of plant as well as in the case of the human. Genome editing is with engineered nucleases. We call it as gene. It's a type of genetic engineering in which the DNA is inserted, replaced or removed from the genome using artificially engineered nucleases. We call them as molecular scissors. This nucleases created a double strand breaks at the desired location in the case of the genome, and we can harness the cell endogenous repair mechanism to induce the break by induced break by natural process of homologous recombination or non-homologous rejoining method. Rejoining both the broken ends is often accompanied by loss or gain of some nucleotide. Repair in non-homologous end joining, thus the outcome of NHEJ is variable. Whereas homologous recombination, the repairing the DNA as a template restore the double-stranded break. Outcome of this kind of repair is precise and controllable. Why we have to do genome editing? To understand the function of the gene or a protein, one interferes with it in a sequence specific way and monitors its effect on the organism. In some organisms, it is difficult or impossible to perform site specific mutagenesis, and therefore more indirect methods must be used, such as silencing of silencing the gene of interest by short RNA interference. We call it as some of the genes can be silenced with the help of a short interfering RNA. We call it as siRNA, so that we can silence the function of the particular gene. But sometimes the gene disruption by siRNA can be variable or incomplete in nature. The nucleases such as CRISPR can cut at any target positions in the genome and introduce modification of the endogenous sequence for the gene. So, requirement. The homing device for specific identification of the target sequence. We have to select the sequence first, then we have to go for the endonuclease function that creates a double strand cut with the help of CRISPR technology. What is the uses of this gene editing process means? To create gene knockout, to find the function of the gene or gene knock-in. As if it is an important gene, when we do it knockout means the gene will be defective, gene can be converted to silent form, so that we can find out what is the function of it. And again, in a controversy, gene knock-in is utilized. Gene tagging, we can tag a particular gene, or we can specifically insert or delete nucleotides for the mutation purpose, and we can see the promoter study also, how the gene is controlled by the promoter, regulator and operator. So novel tools for gene editing includes meganuclease, zinc finger nuclease, TALEN and cluster R, that is CRISPR Cas9. The transcription activator effector nuclease, we call it as TALEN, and clustered regularly interspersed short palindromic sequence. CRISPR, C means clustered, R means regularly, I means interspersed, S means short, P means palindrome, R means repeat. So first and foremost editing tool is nothing but gene editing tool is used for create is meganuclease. The first tool used for double stranded break is induced genome manipulation. So it occurs naturally in the case of the yeast as well as Chlamydomonas. This enzyme binding site and restriction site occurs within the same unit, that is cutting region and binding region. Both are found in the case of this in the same enzyme, so that it can, hence it's difficult to modify it. So crop which is used for conversion, the gene, this meganuclease is used to edit maize and cotton. The maize is made herbicide resistant and cotton is made herbicide resistant and insect resistant. But it is difficult to manipulate the DNA binding site in the case of the meganuclease enzyme. The second one is zinc finger nuclease. These are hybrid resistance enzymes. It consisting of two parts. It is generated by fusing zinc finger DNA binding domain to the DNA cleavage domain. Here we can see the structure of this. The active site contains a zinc that is in turn coordinated with the various amino acids. So we call it as the DNA first. This was the first identified DNA binding motif in the transcription factor, that is TF3A of from the African clawed frog, that is Xenopus levis. Only this TF3A contains zinc finger protein. A small protein structural motif is characterized by this coordination of the one or more zinc ions that is stabilized in this fold. This region is consisting of more than one zinc atom is present that coordinates and the various amino acids present in that region. It contains multiple finger-like protrusions that make tandem contacts with the targeted molecules. And fork1 is naturally found in Flavobacterium echinocoides. So this N-terminal binding domain and non-specific cleavage domain, N-terminal region contains binding domain and a non-specific DNA cleavage domain occurs at the C-terminal. You know very well, N-terminal is amino terminal of the protein and C-terminal is carboxy terminal region of the protein. So development of zinc finger nuclease. Zinc finger domains can be engineered to target the specific DNA sequences so that the zinc finger nuclease can bind to the unique sequences within the complex genome. By taking the advantage, we can create endogenous DNA repair machinery for this. So we can make it as it so that it can bind to the specific region and it will create repair. So the reagents can be used precisely to alter the genome of the higher organism. Here we can see that the binding of zinc finger nuclease to DNA. Here it goes and binds. It restricts the DNA. It cuts the DNA. It cuts uh, the cut sequences may be deleted and new sequences are added to this position so that the break on end are then sealed by its own repair mechanism. Normally, maize, soybean, tomato are modified by this method. Maize has been made herbicide tolerance and soybean physiological trait. We can change it, and again in tomato we can modify it against TYLCV, that is a virus. So it's what is that limitation means? It is off target effect. It sometimes it doesn't lands at the correct position. It lands at somewhere else in the case of the uh, binding position. Construction is cumbersome and it is more time consuming in this case. Another one is TALEN, transcription activator effector nuclease. TALEN means transcription T, A stands for activator, L stands for like, E stands for effector, and N for nucleases. It consisting of tail plus endonuclease. It was identified in the case of Xanthomonas oryzae. The TALEN are acting as a restriction enzyme that can be engineered to cut at the specific sequences of the DNA. They are fusing a TAL effector that binds to the DNA domain and cleavages the DNA by its nuclease action. The transcription activator effectors can be engineered to bind to any desired DNA sequence so that when we combine with nuclease, it can cut at specific location. The TAL effectors are proteins that are secreted by Xanthomonas bacteria by type 3 secretion system when they infect various plant species. These proteins can bind to the promoter sequences so that we can alter the promoter sequence or edit in the promoter sequence of the host plant and we can activate them. Here we can see that molecular structure of the effector. TAL is organized by 3 sections, N-terminal and C-terminal domain. N-terminal domain contains a type 3 secretion signal and C-terminal domain contains a nuclear localization signal and a acidic activator amine domain region, and center region is a central repeat domain and that determines the DNA binding specificity. Nearly a stretch of 34 amino acids found in this region that can be which can bind with the DNA specific region. So in each repeat amino acid, the position 12 and 3 varies. Thus forms a repeat variable residues. We call it as RVD. Repeat variable diresidue. So once the DNA target is identified, the RVD of each target base is selected according to the following code: the DNA base and the amino acid in the tail region, so that it creates a cut in the DNA and it is responsible for editing in this process. So there are different ways by which TALEN can be used to create a disease resistant plant. The transcription activation of the different R genes and transcription repression of different susceptibility gene, mutation in the promoters of susceptibility gene, gene replacement, destroying the pathogen genome. Here is an example of targeted gene modification by TALEN in the plant. And next one is CRISPR-Cas system. These are all the part of the bacterial immune system which detects and recognizes the viral DNA, that is bacteriophage DNA or foreign DNA, and cleaves it. So this CRISPR is clustered, regularly interspaced, short palindromic repeats. C stands for clustered, R stands for regularly, I stands for interspaced, S stands for short, P for palindromic, R for repeats. This CRISPR-Cas9 is associated proteins can target and cleave the invading DNA in a sequence specific manner. A CRISPR array is composed of series of repeats interspaced by spacer sequences and are acquired by the invading genome. Here is the history related to our CRISPR-Cas9 sequence. It was identified in E. coli bacteria but do not characterize their function during the year 1987. Then in 2000s, there was a breakthrough. The CRISPR sequence was found to be common in other microbes also, and they coined the term CRISPR name during 2002 defined the signature of the Cas gene, and first experimental evidence was given during the year CRISPR adaptive immunity, and later they demonstrated the engineering uh, process of the eukaryotic cell by this CRISPR. So here we can see the components of the CRISPR, that includes PAM, that is post spacer adjacent motif, CRISPR RNA, cRNA, and trans-activating RNA, we call it as track cRNA, track or RNA, track or RNA. Here we have the different types of CRISPR proteins are associated with this function from Cas1 to Cas9. Each one is having its own function. You can read the table. Protospacer adjacent motif. It's a DNA sequence that is immediately following the DNA sequence is targeted by Cas9 nuclease and the CRISPR bacterial adaptive immune system. PAM is the component of the invading virus or plasmid, but it's not the component of the bacterial CRISPR locus. The PAM is essential targeting component which is not found in the case of the bacteria. So by using it, it can able to differentiate its own DNA from the invading foreign DNA or bacteriophage DNA. Thereby it prevents the CRISPR locus being targeted and destroyed by the nuclease. Here we can see that the structure of cRNA. Trans-activating cRNA, track cRNA, track or RNA, trans-encoded small RNA with 24 nucleotide compounds is complementary to that of the repeat regions in the case of the cRNA precursor transcript. It pairs with the cRNA, and it is matured by the RNase 3. Then activated cRNA guided by the cleavage of Cas9. So what are all the actions of this CRISPR in bacteria? It is acting as an immune system that protects the bacteria from the virus attacks by the three basic steps, that is adaptation, production of cRNA, targeting. Here we can see the mechanism, how the phage is infecting its DNA, then how the Cas genes gets activated, and cRNA biogenesis and processing, and type 1, type 2, type 3 cRNA is synthesized, then cascade complex, and it identifies the target and causes degradation, in which we can see the role of PAM, Cas3, Cas9, and CAM complex, sgRNA, and CMR complex. It's a versatile nature of the CRISPR technology, so which is in turn used as a tool to not only edit plant genome but also human genome also. Here is the general protocol for the CRISPR. So this is the advantage of our CRISPR technology. This is an application of the CRISPR Cas9 in the agriculture. There are some pitfalls in the case of this technology. There is a limited choice of the BAM sequence, and proper selection of the gRNA is very essential. And Cas9 Cas9 version varies. We have to ensure that there is no mismatch in the sequence of this. So by using these techniques we can modify the plants instead of the normal plant breeding method, which can improve our yield and quality, and further it provides disease resistance to the plant in this case. That's all about the tale of your CRISPR Cas9 zinc finger nuclease and TALEN's technology in gene editing process. Thank you.